Okay, so when we're considering the synapse, the target-rich site for drug interactions, and also where, the action potential, right, the electrical message, the depolarization and repolarization is arriving and being converted, into a different language, the release of a chemical neurotransmitter, usually more than one, however, usually a primary neurotransmitter. That's the majority of what's contained in the vesicles, for release upon the arrival of an action potential. We need to start talking about what the different protein targets would be for each of these different types of neurotransmitter. Because I mentioned earlier we have dopaminergic cells, right? Those are neurons that are going to primarily synthesize, store, and release the neurotransmitter dopamine. But there's other neurotransmitters too. And there's a whole sort of class or a designation for a set of neurotransmitters that we refer to as the classical neurotransmitters. And the classical neurotransmitters, in this context, the word classical just means early or historical. These are the neurotransmitters that were identified or discovered first. And the very first neurotransmitter that was discovered, and we'll go into some detail on it, is one called acetylcholine. And there's a famous story of Otto Lowy, who stimulated the vagus nerve, which is the primary sort of parasympathetic, output to the upper viscera. Remember the parasympathetic division of the autonomic nervous system, which serves to slow down the heart rate. And he, had a beating frog's heart, in a dish with the parasympathetic nerve, the vagus nerve still attached. And he stimulated it, and the heart slowed. He ended up then, taking a little dropper and dripping the fluid, after he had stimulated and slowed the heart onto another beating frog heart in another dish, and it also slowed. This actually was one of the ways we, figured out that the neurons communicate with each other, in terms of chemical connections, as opposed to just sort of direct electrical links. Acetylcholine is probably the most classical of the classical neurotransmitters. It was initially referred to by Otto Lowy, this Austrian neuroscientist who did the frog heart experiments, as vagus stuff, like literally the stuff that came out of the vagus nerve. But it turns out it is, the primary neurotransmitter for the parasympathetic division of the autonomic nervous system, which is acetylcholine. Then there's another class of classical neurotransmitters that play a pretty significant role in drug use disorders and just in normal motivated behaviors. Right? These are called the monoamines, and the monoamines we're going to see refers to the fact that they have one amine group, and it's an NH2 group attached, and they're all, it turns out, manufactured from amino acids, which are the building blocks of proteins. And two of these, dopamine and norepinephrine, these are two monoamines that are actually made from the same amino acid. It's an amino acid called tyrosine. And because norepinephrine and dopamine are derived from the same amino acid, it turns out they share additional structural features in addition to the single amine group, the single NH2 group. They're not just monoamines, they're also what we call catecholamines, because tyrosine, which is the raw material to build both dopamine and norepinephrine, actually has a distinctive sort of chemical feature. It's what's called a catechol ring, and that's preserved in both dopamine and norepinephrine, which are manufactured or synthesized, from the amino acid tyrosine. So the monoamines consist of dopamine and norepinephrine, right? These are the monoamine neurotransmitters. These are classical neurotransmitters. They were discovered and identified a long time ago. Dopamine and norepinephrine, which are made from an amino acid called tyrosine, share certain structural features. So collectively, dopamine and norepinephrine are called the catecholamines. That's a subset of the monoamines. And the other critical monoamine, that's one that I know you've heard of and is a very important classical neurotransmitter, is serotonin, which also has an NH2 group, so it's a monoamine, just one NH2 group, so it's a monoamine. But it also has a different kind of a ring structure. It's called an indolamine, because it's derived from a different amino acid raw material. It's actually made from the amino acid tryptophan. So The monoamines consist of dopamine and norepinephrine. Those are the catecholamines that are derived from the same, synthesized from the same amino acid, which is tyrosine. And then there's serotonin, which is also a monoamine, but it's a different kind of monoamine from the catecholamine because it's made of a different raw material, the amino acid tryptophan. We'll talk more about this as we continue with the class. Then there are sort of, sometimes people refer to them as the amino acid neurotransmitters. These are neurotransmitters that are very close to, amino acids that we consume on a regular basis. One is called glutamate, which is made from an essential amino acid, which is called glutamine. And then there's GABA. And in some cases, GABA is actually made from glutamate. And what's interesting is that while the monoamines, get a lot of attention because they're involved in motivation and, psychological health and arousal, mood states and things like that. The amino acid neurotransmitters, glutamate and GABA, are kind of the workhorse neurotransmitters. They're far more, present at more synapses, throughout the central nervous system. And we're going to learn that glutamate is the primary excitatory neurotransmitter in the brain. And what I mean by that, of course, which you should now know, right, is that to excite, you depolarize. So it suggests that glutamate will have to bind to some kind of a postsynaptic receptor that's going to result in the opening of some kind of a depolarizing current. The opening of a sodium channel, for example. Sodium will come in, driven by the electrical and concentration gradient forces, or opening a calcium channel or a sodium and calcium channel, right, to drive positive ions inside and depolarize the cell and move it towards the threshold for initiating an action potential. GABA, in contrast, is our primary inhibitory neurotransmitter. We'll see more about that too. But GABA, is going to bind to receptors that are going to open some kind of a channel, we'll see, that will result in not depolarization, but hyperpolarization. It'll make, an action potential less likely because it'll move the charge difference across the membrane further away from the, threshold potential of minus 55. It'll go from rest, to something like minus 70 or minus 80. And we're going to see it's going to open, in most cases, like a channel that will allow for the passage of chloride ions from the outside into the cell, which would result in hyperpolarization. So these are pretty much the classical neurotransmitters that we'll be talking about in some detail. Acetylcholine, the vagus stuff, the monoamines, the catecholamines, dopamine and norepinephrine, and also our indolamine or our third monoamine, serotonin. And then the amino acid neurotransmitters, glutamate, our primary excitatory neurotransmitter, and GABA, our primary inhibitory neurotransmitter. Now, there's a whole host of additional neurotransmitters that we'll talk about in some detail as well in this class. For example, these are non-classical. These were identified later. There are what we call peptide neurotransmitters. Peptide means short protein. So it's a short sequence of amino acids, actually. And generally with the peptide neurotransmitters, we're going to see many of these are actually opioid neurotransmitters, which are going to play a significant role in inhibiting information about injury in the body, from reaching maps in the central nervous system. A lot of drugs obviously target the opioid system, but peptide neurotransmitters are often, there's a gene that'll express like a larger protein in a neuron that's going to release opioids, synthesize, store, and then release, and then degrade the opioids at a synapse. Actually, peptide neurotransmitters actually are often co-localized neurotransmitters. There's often a primary neurotransmitter like dopamine that could be co-localized with a secondary peptide. But the peptide neurotransmitters, there's a gene typically in your nucleus of the cell that will release them. That'll code for a bigger protein, and then other genes will be expressed also in a neuron that'll release a peptide that'll actually serve to, they're enzymes that'll serve to cut small segments, basically out of that larger protein that you've transcribed and translated from a gene. And then, all the other machinery for packing it into vesicles and then release and then removal actually will also have to be expressed as well. So peptides are a sort of non-classical, neurotransmitter class, and there's quite a number of peptides that have been identified. Then there's also lipids, and these are fats, and there's a very important set of lipids that you actually manufacture, and that drugs certainly impact. These are the cannabinoids. And we'll talk more about this later, but there are cannabinoids that you generate in your own brain. They call them endo, because that's body, your own body, endocannabinoids. And these are fascinating neurotransmitters because, for one thing, you can't store them like you can all the other neurotransmitters we're discussing in a vesicle because they're fats. So remember our phospholipid bilayers. Well, they can just drip right out of them. They can get right through them. So they can't be stored in the same way. We're going to see they're actually typically stored in the postsynaptic membrane at synapses, and we'll talk about this later when we talk about cannabis. But in the postsynaptic membrane, they have to be kind of cleaved out or cut off other structures that are present in that membrane so that they can then drift into the synapse and then stimulate receptors that are often kind of interestingly presynaptic. So cannabinoids are really interesting. Lipid neurotransmitters are a very non-classical category of neurotransmitter. We'll be talking about all of these later in the course.